if you think of life, it's an orchestration of molecules. Life has somehow decided that these molecules have to move in certain ways, for cells to move, for muscles to contract, for microbes to live in the ocean in conjunction with corals and to branch out and deposit silicates. And so if you think of it, what mass spectrometry is really doing, it's capturing life, and it's doing so by weighing how heavy the molecules really are, and that translates in us being able to develop a picture of what is really present in a given biological sample.
People may be working on a molecule that's very interesting, and they spend a lot of time figuring out what that spectrum means and how to interpret it, and they put labels on every single peak. But then at best, it ends up in supplementary materials to a paper that is not searchable, is hard to find, and it's next time the same molecule shows up in anyone else's data, they're just going to have to do it again from scratch. And this is not a simple job. This is a highly specialized job. In certain classes of molecules, only certain labs are able to deal with that in a reasonable amount of time. So because we don't have a way to connect these experts to reuse this work and this knowledge, a lot of things have gone unidentified in mass spectrometry.
Some of the things that we can do is we can compare a soil sample that maybe somebody on the other side of the planet has collected, and we have a lung sample, and we want to see how many natural products, how many of the molecules are the same, and how many are different. Before Gnps, there's really not a good infrastructure to make that type of correlation.
If you think of how genomics came to be this fundamental pillar of biology research in the 21st century, it really was the availability of algorithms to put together whole genomes from small DNA reads. There's this fundamental principle in genomics that sequence similarity implies functional similarity. That if biology creates a genomic sequence that is identical, very likely to function, if any, of that sequence will be similar. So what we have developed is essentially the equivalent algorithmic foundation for mass spectrometry, where what we look at is if two molecules have identical or similar fragmentation patterns in mass spectrometry, then what we've observed is that usually corresponds to structural similarity of the molecule that generated the spectrum. So now, by developing algorithms that process spectra directly and match spectra to one another, we can start to find molecules that are related to one another. And if they're not related, then their spectra will not be related either. So it gives you a tool to explore the molecular space as observed by mass spectrometry in the same way that genomics gives you a way to explore the space of genes and RNA and everything that's coded in the genome in an unbiased manner, right? You don't have to know what is the function of the sequence before you can detect that there's sequence conservation, and then using that conservation to guide the research forward. So what this allows us to do, these algorithms that we have in a GNPS platform, is to take the same kind of unbiased approach to the mass spectrometry world, to start finding spectra of related molecules that may be variants of something that is already known, or they may be completely new molecules.